Hello and welcome to Ryan Hennessy Wastewater Microbiology YouTube channel. This video, I'd like to just do a quick walkthrough of the DNA to the micro DNA summary report. So basically, the way this works is I receive a sample, create a micro report, which is sent to the client, and I preserve the sample, send it to the DNA lab that I work with, and they give me back a report showing the DNA sequencing reads, which is using 16S RNA sequencing. What I do is I take their results and I compare them to the microscopy, and I look for the correlations, and we have a growing database of over 150 samples at the time of this recording, where we're getting some correlations in addition to other known correlations of morphotype through the microscopy versus what you would see with the genus in the DNA read. So basically, I get this report. I use AsterBio for the DNA lab, and the particular sample that this was taken from had very abundant to excessive type 021 filaments. Type 021 filaments are commonly associated with the Thiothrix genus. You can see here the reads of Thiothrix are very high. They have a nice setup with the way that they display their reports. What I generally look at here is I filter through here, and I'm going to sort these or filter these from highest to their largest to smallest reads. So then that's going to show me, and I'll compare that to what I'm seeing on their functional groups tab. Then what I do is I copy the chart from my microscopy report, and here you can see the different filaments and their abundance. So Zuglia was ranked abundant or very common, type 021N, abundant. Some Thiothrix filaments, Flexibacter, and so on. So I cross-reference looking at the DNA report, and Thiothrix genus was 33.7% reads. 021N filament was there. It was abundant. So obviously very high. That matches very well. And Thiothrix can be either of those morphotypes. Same with Zuglia. We had a very high, fairly high rate of Zuglia bacteria, Zuglia morphotypes, which also can be Theora here, which is another genus that we saw of note on the DNA test. Flexibacter are very diverse from a morphological standpoint. In this instance, there was a unnamed genus that is in the same family as a common known Flexibacter morphotype. So, I always put in a little disclaimer that some of these are not exactly 100% proven. However, a lot of times if there's similarity between the families or things like that, or if it's just something that I see with a fairly high correlation rate, it's something that I note as a potential candidate. So the DNA also tells us, and in this example, adds to the microscopy from the standpoint that there are very low percentage of nitrifying bacteria reads. So Nitrospira, Nitrosomonas, both very trace reads. Generally in a municipal wastewater plant, about 1% of nitrifying DNA reads would be fairly representative of a stable nitrifying population. However, that's going to vary a lot depending on what the BOD to N ratio is. So there were a couple other findings from the DNA, a couple other genus that could potentially be filaments, and they were all fairly low percentage. For example, Runella genus is potentially could be a 0411 filament type. It can also be variable. So a lot of the different filament genuses can be either present as filaments or single cell or tetrads or other forms. So, there was a trace amount of spiralis, which can also be single cell um, in addition to filaments. So it does give us a little bit of extra information. And the other thing that I do is I will reference the MIDAS database, which we use for all of our DNA sequencing reads. And anything on the MIDAS database can be searched here through the field guide. So for example, you can look at Thiothrix, can look that up, and it's going to tell you, right now there's 46 species recognized and other information about Thiothrix. So basically what I'm doing with the DNA micro is I'm taking this data from the DNA company along with my micro and kind of summarizing the micro or the report of the DNA to something that's hopefully of value and can be digested fairly easily. So then I have a common kind of a summary of what I saw, what the correlations were. Then there's also a little disclaimer as we talked about, and that's another separate topic as far as, how interpreting DNA reads and things along those lines. So anyways, just wanted to share that with you. This is the DNA to microscopy evaluation report.